sort of the whole next generation of iPS cells where they're using non-viral ways of getting the DNA in there. Sometimes it's RNA, sometimes it's protein, sometimes it's just in plasmids that won't integrate. So those are hurdles that are being overcome. Another question is, are these really embryonic stem cells? So just because it looks like an embryonic stem cell, can you prove that it really has all the properties? And depending on who you talk to, people really are not of a like mind on this. Some people absolutely think these are bona fide embryonic stem cells. Other people argue they're not. I don't know what it would take to convince either side of the other, so I think this is just an ongoing issue in science. So this is a slide that Yamanaka produced for his Nobel lecture, and I just really liked it because it highlights the streams of science that all came together, in this case for his work. But so I already told you about Gurdon and the predecessors of Gurdon, and Wilmot cloning the sheep, and then we talked about embryonic stem cells and human stem cells, and we talked about MyoD, and all these things together allowed scientists to come up with this idea that iPS cells might work, that you could actually reprogram a cell based on a very small number of factors, and that you could have it have the potential of these embryonic stem cells that people had worked. So I think the way he brings it all together is really nice, and what I'd like to sort of finish up with is where this might go. So what is the future of this research? And this slide, in a nutshell, is really everything that are the big questions for iPS research. And the fundamental idea behind it is that you can take cells from an individual, here, like a patient, and you can make iPS cells, and those iPS cells then can be called